Hi, my name is Mr. Evangelista, and today we're going to be talking about genetic engineering. So in my past couple videos, we've talked about DNA and what DNA does and how DNA gets changed into RNA, into protein. So you know all this stuff about how, genetic expression works, but I'm sure the question you've been asking is, what can I do with this information? More importantly, can I manipulate DNA to express certain traits that I want? And that's what genetic engineering is all about. It's all about manipulating DNA, transferring DNA maybe from one organism to another to transfer specific traits that we might like. And so that's what we're going to be talking about today. And so let's start off with an example. Let's say I have a cell, and inside this cell has a specific trait that I want. We need to somehow take out the DNA from this cell, be able to examine the trait that we want. So let's go ahead and do that. And the way we do that is with a solution called lysis. What lysis does is it's going to break the cell membrane and the membrane around in the nuclear membrane, releasing all of the contents of the cell. Now, when we have all the contents of the cell, we need to somehow separate these contents from the DNA itself, because we're going to have the mitochondria in there, the Golgi apparatus, a bunch of proteins, and we don't want all that stuff. All we want is the DNA. We would perform a technique called DNA extraction. Simply, what DNA extraction is it gets some salt, some alcohol or ethanol, and we put that all inside of a test tube, and we're going to put them inside a machine called a centrifuge. What the centrifuge does is it's going to spin these contents really quickly, and it's going to separate them according to their densities. And so once we've gone through this process of DNA extraction, we have our DNA. Now, this DNA, we only want a specific section of the DNA. So let's say we are looking for the section of DNA that codes for the production of insulin. Okay, so if we know where that section is and we know how long it is, then what we can do is we need to cut it out. And the way we do that is we use these things called restriction enzymes. Now, what restriction enzymes do is they cut DNA, but they'll cut them in a very specific way. Each restriction enzyme has a specific sequence that it will cut at. And so this restriction enzyme that we're looking at here happens to be called EcoR1. And if we look at this DNA sequence here, it will cut along the DNA wherever it reads this specific sequence. And so if we look at our DNA molecule, we can see that it would cut this DNA molecule in these 3 parts. And we would therefore get 4 fragments. Now, only one of the fragments is our target fragment, meaning the fragment that we want. We can use a process called gel electrophoresis, and that uses this agarose gel. It's kind of like a jello, and it's made from seaweed extract. And how this works is we can put all of those fragments inside this top little black well, and when we run electricity through this, the fragments will go down the gel, and in smaller fragments will travel faster and further than bigger fragments. And so we can use this process called gel electrophoresis to separate these fragments by size. We have our target fragment. We can make a bunch of copies of it, and we do this through a process called PCR, or polymerase chain reaction. Now, the reason we would want to do PCR is all of those steps that we just did to extract our target gene, those were a lot of steps. And if we want to study a gene many, many times, we don't want to have to keep going through that process over and over and over again to get multiple copies of our target gene. So instead of doing that, we can use PCR, and what PCR does, it makes many copies of our target gene. And so now that we have a lot of copies of this gene to work with, we can put it inside what's called a plasmid. Now, what a plasmid is it's a circular piece of DNA that's used in bacteria. And so what we can do is we can cut that plasmid with the same restriction enzyme that we use to cut our original DNA. The reason this is important is because if we cut it with the same restriction enzyme, then the ends of those DNA are going to be they're going to be complementary to each other, meaning they can stick back together. And so what we can do now is that we have a cut plasmid, we have our target DNA, and we want to glue these back together. And the way we can do that is with an enzyme called DNA ligase. And what DNA ligase does is it helps put back together those covalent bonds in between those DNA molecules. Now, there is a problem, and as you're going through this, you should be coming up with some questions about the processes we're going through. Ideally, what we want is we want our gene to be glued right in the cut we made in the plasmid so that we get a plasmid with our target gene in it. Now, this doesn't always happen. In fact, what we could have is we could have the plasmid that just seals back on itself. We could have just a bunch of target genes attached to each other and not attaching to a plasmid. We could have a plasmid with several copies of our target gene in it. And so we can see that I have a lot of different options here. And what I want you guys to do is kind of think of ways that you could isolate just the plasmid with one copy of the target gene in it. What are some techniques or things that we've gone over in class that you could use to isolate just the plasmid with one copy of your target gene? Think of some answers to those. Maybe you can comment it down below, but we're going to go ahead and move on. And so we can take this plasmid that we have with the target gene in it, we can actually put it inside a bacteria. Now here's a bacteria. Bacteria have their own DNA, but they can also accept plasmid DNA. Now how we get this plasmid inside the bacteria is we're going to have to shock it. We get these tiny little pores that open up in the bacterium. And once those pores are opened up, then we can put the plasmid in the same solution as the bacterium, and hopefully some of those plasmids will make it inside of that bacteria. And there we go. We have just created an insulin-producing bacterium where we took the genes from an insulin-producing cell and we have inserted it into a bacteria, and now that bacteria is programmed to create insulin.